Thank you for your interest in Vet NeuroJar. Although we do our best to summarize veterinary articles, it does not supplement the actual article, and if you are able to download and review the article yourself, please see the link in the description. This will summarize the article titled, Epilepsy is More Than a Simple Seizure Disorder, Causal Relationships Between Epilepsy and Its Comorbidities. Epilepsy is a brain disease that results in epileptic seizures, along with social, cognitive, and neurobiological changes. The cause and progression of epilepsy, known as epileptogenesis, and its comorbidities are being investigated through various clinical and preclinical studies in humans and laboratory animals. Anxiety, cognitive impairment (CI) and attention deficit hyperactivity disorder, ADHD, like behavior were discussed in a recent review as the most prevalent behavioral comorbidities in canine epilepsy. This review focuses on the pathological mechanisms that link epilepsy to its comorbidities. Pathological mechanisms such as anatomical structures, functional connections, neurotransmission, and inflammatory processes can all contribute to the etiology and progression of epilepsy and its comorbidities. The link between epilepsy and its comorbidities is complex and may involve bidirectional mechanisms, shared pathologies, and coincidences. Many people with epilepsy often experience comorbid psychiatric conditions such as depressive disorders, suggesting a bidirectional relationship between these conditions. The relationship between epilepsy and its comorbidities needs more research in dogs. This review presents an overview of the possible mechanisms linking epilepsy and its comorbidities in canines based on clinical and preclinical studies in humans. To gain a better understanding of the complex relationship between epilepsy and its comorbidities, researchers may analyze the anatomical structures involved in focal epilepsies. Structures within the limbic network, such as the amygdala, hippocampus, and entorhinal cortex, are potential sources of seizure generation. The amygdala is a key structure for experiencing and mediating fear within the brain, and an increased excitatory output from these neurons may lead to anxiety states. Clinical and preclinical studies on rats have demonstrated that electrical stimulation of the amygdala can lead to seizures and anxiety. Recurrent seizures emerging from limbic structures may function as endogenous stimulators of comorbid interictal anxiety, hence the interplay between anxiety and epilepsy involving the amygdala appears highly plausible. The hippocampus is frequently identified as a seizure-initiating area and is involved in functions such as memory, cognition, and fear. Hippocampal sclerosis is the most common neuropathological feature of EPLE in humans, leading to loss of neurons and reduced volume that may cause or result from epilepsy. Cognitive impairments may occur independently of epilepsy. Hippocampal structure alterations, such as HS, may present a common cause for epilepsy and cognitive impairment. Lastly, studies have identified brain structure differences in patients with epilepsy and comorbidities, such as ADHD, suggesting various pathological mechanisms leading to varying combinations of these diseases. Functional brain networks may be responsible for the associations between epilepsy and its comorbidities. Both epilepsy and affective disorders disrupt neuronal circuitry. In human medicine, electroencephalography (EEG) and/or functional magnetic resonance imaging (fMRI) can be used to determine seizure focus localization and functional connectivity (FC) of brain regions. However, using fMRI and EEG in dogs has its challenges and practical issues. For example, fMRI requires weeks of training to keep dogs motionless in a scanner, which may be difficult for dogs with epilepsy and anxiety or related behavioral disorders. EEG examination is also challenging in dogs due to varying skull size structure, brain size, and large temporal muscles, FCS, are commonly altered in seizures in parts of the cortical and limbic system that mediate cognition and memory. Disruptions in the prefrontal limbic networks and temporal lobe epilepsy, TLE, that are associated with cognitive dysfunction are also linked to depression. Interrupting or altering these FCS within the epileptogenic brain areas might have anti-seizure effects and suppress comorbid mood disorders. There is evidence that interictal epileptiform discharges, IEDs, detected by EEG, cause and precede brain network changes in children with epilepsy and dogs with IE. Furthermore, IEDs can impact cognitive decline, anxiety, and depression even in patients with benign Rolandic epilepsy. However, a definitive bidirectional relationship has not been established in terms of functional connections. This suggests that epilepsy and its comorbidities may have a shared pathogenesis, as similar brain areas are affected in cognitive and mood disorders independent of epilepsy. The role of neurotransmitters in epileptogenesis and comorbidities has been extensively studied. 5-hydroxytryptamine, 5-HT, which mediates the effect of GABA and glutamate, is suggested to play a role in epilepsy. Mimicking 5-HT and selective serotonin reuptake inhibitors exert an anticonvulsive effect. Decreased 5-HT1A receptor binding has been observed in anxiety-related and depressive disorders. GABA, the main inhibitory neurotransmitter in the central nervous system, is also involved in epilepsy and epileptogenesis. Epilepsy-induced neuronal cell loss in the hippocampal formation present compensatory mechanisms in the epileptic brain. Benzodiazepines, which enhance GABA, reduce central excitability and exert anti-seizure and anxiolytic effects. Noradrenaline, a neurotransmitter in the central and peripheral nervous systems, is also suggested to be involved in ADHD and epilepsy. Genetic epilepsy-prone rats with noradrenergic deficits have increased seizure susceptibility, and a substitution of NA decreases seizure vulnerability. Epilepsy and ADHD might be connected by reduced levels of NA. The pathways of these neurotransmitters in affective disorders and epilepsy seem to be closely linked, influencing each other. Chronic inflammation affects various brain functions involved in epileptogenesis, such as neurotransmission, neuroendocrine function, blood-brain barrier integrity, neuronal excitability, synaptic plasticity, neurogenesis, and astrocyte function. According to research by Causal et al. 2018, the role of inflammation as a causative or consequential factor of epilepsy remains unclear, as reported by Vesini et al. 2011. Nonetheless, increased levels of pro-inflammatory cytokines including interleukin
È una vita.
IL,
È una vita.
Il 1 Ra, il 6, il 17, tumor necrosis factor.
Alfa. TNF, alpha,
and transforming growth factor,
beta,
TGF,
beta,
have been detected in the blood, cerebrospinal fluid, and surgically resected brain tissue of individuals with epilepsy. Hulkinen et al., 2004, Ledemaki et al., 2007, Sinhai et al., 2008, Novak et al., 2011, Strauss and Ilisevich, 2016, Arend et al., 2018, cyclooxygenase 2, COX2, and prostaglandin E2. PGE2 expression and the production and activation of inflammatory mediators have also been observed during seizure development and progression. Wang et al., 2018, Singh and Goel, 2021. These inflammatory mediators alter neuronal excitability by modifying neurotransmission of glutamate, GABA, serotonin, and ion channels. Podol et al., 2018. Suleimanova, 2021. Chronic neuroinflammation leads to hyperactivity of the hypothalamic pituitary adrenal axis and upregulation of 5-HT1A autoreceptors, which results in decreased Raphe hippocampal serotonergic transmission, causing a serotonergic deficit that may contribute to seizure occurrence. Pineda et al., 2010. This may also explain the co-occurrence of mood disorders and neuroinflammation, due to the effect of decreased serotonin concentration on mood. Neuroinflammation is associated with mood and cognitive disorders including depression, anxiety, and cognitive decline. Vogels Angs et al., 2013. Slavich and Irwin, 2014. Marsland et al., 2015. In epileptic individuals, as shown by Barker Heliski et al. 2017. Mazzarati et al. 2017, Singh and Goel, 2021, and Suleimanova, 2021. One study found that epileptic individuals with depression had higher plasma IL
ένα β,
concentrations that correlated with the severity of depressive symptoms compared to individuals with epilepsy, but without depression in healthy individuals. Vieira et al., 2015. Anti-inflammatory treatment in a rat epilepsy model has shown a reduction in seizures, depressive symptoms, and brain inflammation. Shen et al., 2019. Long-term inflammatory mediator activation contributes to neuronal dysfunction and cognitive deficits in learning and memory. Cunningham and Anderson, 2008. Reactive oxygen species during chronic inflammation and recurring seizures cause oxidative stress accompanied by mitochondrial dysfunction, leading to neuronal cell death and cognitive decline. Revisa et al., 2017. Diavola et al., 2018. Mitochondrial dysfunction affects neuronal calcium balance and reduces ATP concentration, compromising synaptic transmission and neuronal excitability, increasing susceptibility to seizures, and exacerbating mitochondrial damage during ensuing seizures. Patel, 2004, Rahman, 2012. Interestingly, medium-chain triglyceride-enriched diets have reduced seizure frequency, cognitive decline, ADHD-like behavior, and anxiety in dogs with IE and individuals with epilepsy by enhancing mitochondrial function, confirming the involvement of mitochondrial dysfunction in chronic inflammation in epilepsy and ADHD, as indicated by human studies. Yaf et al., 2004, Anand et al., 2017. In summary, inflammation presents a significant link between epilepsy and its comorbidities, with multifactorial characteristics influencing disease development and emergence. This review delves into the interconnections between epilepsy and its comorbidities, such as abnormal neurotransmission, neuroinflammation, and the limbic system. These factors are instrumental in the onset and progression of epilepsy and its associated conditions. Though the hippocampus and amygdala are known to play a role in seizures, they also function in cognition, memory, anxiety, and depression. Changes in these regions, such as degeneration and inflammation, impact functional connectivity and neurotransmission, and influence clinical outcomes. The outdated hypothesis that epilepsy is solely responsible for comorbidities has given way to the common cause theory, which recognizes that multiple factors contribute to a shared pathogenesis. In the case of canine comorbidities have only recently emerged as a focus. Therefore, further research is required to understand the relationships between epilepsy and psychiatric and cognitive disorders, and how best to treat and manage them.